guys, let's see. Oh. Yeah, let's get started. So today we will start talking about cell culture techniques, especially in neuroscience technology. I have a little bit of a material to cover, so bear with me, and I think we'll have enough time to go over. So let's see. So that's, I want to put in the bottom. Okay, so today's goal is comparing in vitro culture techniques and in vivo techniques. And we want to describe types of cells used to examine a nervous system function, especially in vitro, because we are talking about cell culture techniques. The techniques we will cover today is tools and reagents used in culture. Culture is not like Munha. This culture is about growing cells and tissues on a dish. So we will talk about equipment and media. So types of cell cultures, about 3 types of cell cultures were discussed today is immortalized cell lines, primary and tissue culture. Let me see. Okay, let's first discuss, define what is cell culture. Cell culture is the process by which a scientist grows and maintains cells under carefully controlled conditions. And cells are originally inside the body, but we are talking about outside of a living animal. So first, the Latin word we have to define. I'm pretty sure everyone knows. In vitro is literally in Jibam. Can you guess what this is? Oh, yeah, it's a glass, okay? So it's a Latin, so not easy, but just understand one time. In vitro means in the glass test tube, a process performed or taking place in a test tube, either culture dishes or elsewhere outside of a living organism. Then we talk a lot about in vivo. So literally in vivo means in a living thing, okay? Which is already, I wrote it many times, a process performed or taking place in a living organism. So if you look at these picture, this will give you some idea that in vitro assays usually have these like plates, weld plate, and the pictures you are observing is in fact the cells are on top of a plate. So this is more like two-dimensional shape. Looks like you know, egg, right? Egg plate when you are like cooking an egg, and this is a more multiple number of cells. Here it says transformed cell lines. Transform is used when a cell turned into a cancer cell, immortal cells. So they change their characteristic to live forever. So that's why we call sometimes we call cell line, not just cell lines. It has a permanent and almost infinity line lineage. You can keep the cells. So that's why we call cell lines. In comparison, we take out the cells from the animal. You guys did this last week as a practice, right? So those are called primary cells. While those are, we grow the cells outside of the body, but the cells in its native environment, which is, let's say, inside the living body of animal, that we call in vivo assays. So you can see in this case, this particular example is nanoparticles injected into the tail vein so that it distributes throughout the body. And we wonder how these nanoparticles or maybe treatment or these are distributed. So in this case, you can see obviously in the liver, there's a lot of accumulation and maybe sometimes other tumor sites. Okay, so why we are talking about why we are interested in vitro experiments. As you see, you take out to make it simplify the cellular environment. So it should have a greater con over experimental manipulation. Julie, can you guess what this is?
Read the condition. It's condition.
Video editing software.
So it has a greater control over experimental manipulations because now it's much easier to control, okay, the conditions. And we reduce potentially,
like
Drew, can you guess this? This is not easy though. Potentially conding interactions with other biological systems. So in vitro experiment, we can limit the parameters. We can control the parameters carefully. While in vivo experiment, when you do, let's say, giving some treatment or manipulation, you see some outcome. However, in vivo, intrinsically, the interaction of the cells of our interest with the microenvironment are complicated. So we potentially a confounding effect that makes it sometimes more difficult to draw conclusion. So, Dri, can you guess what this is? Reduce potentially confounding interactions. You know, these terms are very, very important to understand, okay? Which means we can try to understand independent variable A affecting dependent variable B, so that trying to establish a causal relationship, A causes B. But sometimes if things are more complicated with many players around, you can have like confounding effect, which makes it more complicated to draw conclusion. So that's why in vitro experiments are simple in that aspect, okay? So of course it's faster than in vivo, less expensive, because keeping the animal and growing, raising, making transgenic animals all are very costly and require fewer animals because you took out a lot of cells from animals. So an example of in vitro experiment is performing multiple assays in parallel with the same number of cells. So let me go back to this picture. You can see this 1, 2, 3, 4, 5, 6 well played. So when we do experiment, we can have some outcome, one outcome, but it's very hard to draw conclusion because in science, we have to repeat the experiment or we have to establish the statistical significance of whenever we see any difference. However, even one animal is hard to deal with, but to achieve some scientific conclusion, we need many, many animals, which is of course much more difficult. In comparison, this in vitro assays, let's say you took out these multiple primary cells or transformed cell lines, you can actually have a precise number, let's say 1.1 million cells over here, 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 simultaneously. Then you do the experiments, then what it means is you can do in one experiment, you can actually have a 6 data set, which will help you to establish statistical analysis. So that's why performing multiple assays in parallel with the same number of cells, which is a pretty difficult job in vivo. Okay, so that's what the merits of in vitro experiments. Then what are the goals of in vitro experiments? Okay, to generate a hype and projection about what occurs in. So Min-sung, can you try what this means?
Hypothesis.
I can't hear you very well.
Hypothesis.
Okay, yeah, I understand. Hypothesis, yes. Projection about what occurs in
In vitro.
Yes, I'm watching your mouth because I can't hear you somehow. But anyway, yeah,
In
you know,
vivo.
our goal is what happens in vivo. That's our ultimate goal. However, in vivo is much more difficult. Sometimes these confounding interactions all makes it very, very difficult. So many times it's, you know, it's a good approach to devise and develop an in vitro assays to generating hypothesis, which is maybe very good to test in vitro condition, but we have to make sure that in the reason why we perform in vitro experiments is about finding what occurs in vivo. That's the ultimate goal. So you have to keep that in mind. A very simple example I can say is you will rarely find in vitro only experiments to publish in Nature Science, okay? Even you did a great idea, great job to prove in vitro, you know, people will ask you, at the higher rank scientific community will ask you, hey, have you seen it in vivo? Have you seen it in clinic? Or what you observed in vitro results happening in real human patients? That's usually what people will ask. So you have to understand, this is ultimately what we are interested, okay? Thus, design in vitro experiments very carefully because you can control very well and extrapolate results cautiously. So don't say to the mass media that your in vitro experiment, you find out, hey, I have found out a good way to kill cancer. And then like say to the newspaper, hey, we figured out how to kill the cancer. You gotta be very, very careful because extrapolation to in vivo, there are many hurdles, such as this confounding effect. Proving in vivo is sometimes very not easy, okay? So you don't necessarily draw conclusion of in vitro experiments, interpret too far. So you always have to confine or the scope when you draw conclusion. This is very important in science. And whatever you do, you have to set the scope to draw conclusion. Don't just say like beyond what your experiment and say this will be applicable to everywhere in vivo in human patient. We gotta be very careful for that. Okay. So cautions for in vitro experiments. Does a cell in a culture flask? So this is about general question when we are performing in vitro experiments. Does a cell in a culture flask truly behave like it would inside a brain or body? I mean, this is the most important question you have to keep in mind when you are running in vitro experiments, which is usually in a culture flask, okay? So it is very good idea for you when you run your experiment, you got results, your interpretation and conclusion, you have to ask yourself, hey, I found a very interesting contrast or differences between the manipulation or not, the difference in the outcome. You have to think this. I would say a general answer for this is we do not know until we actually test it out. That's why in vitro experiments is a good starting point to generate hypotheses for in vivo experiments. Okay, because you don't know. So for example, why we do not know in vitro test results. For example, let's say I'm studying neuronal firing with giving certain different kind of neurotransmitters and or drugs affecting the neuronal firing. However, like think about the in vivo real situation. Neuron never be alone. Neuron will be interacting with a neuroglia, okay, other types of non-neuronal cells, or there are blood vessels, there are microglia, immune cells. In vivo condition is much more complicated. So drawing conclusion is very hard to know until we actually perform the experiments. Okay. And another aspect of the cell itself, what we have to ask ourselves is, are immortalized cell lines? I said cell lines. Okay. That is cells that continue to divide indefinitely. That's why we put it as a line, okay? Which is a good model for cells that degenerate and perish in the brain. So this is about the eligibility of the cell itself. Okay, I think a good example is in not even neuroscience, but the cancer study. Like there are a number of even like tens, dozens of, or hundreds of cancer cell lines. For example, breast cancer cell line, there are dozens of cell lines, but we can't do everything. So we use one or two cell lines to run experiments. And then, hey, we found a good compound that can kill this cancer, breast cancer specifically. But then the conclusion has to be very, very careful because this cell line is coming from one single individual cancer patient. A good example is HeLa cell, which is originated from like HeLa Lex, who is a woman who got a cervical cancer, very aggressive tumor. So there comes out a cell line, which is 70 years ago. So we have been studying using it for a long time. But the cell lines means that has transformed from its original into adapting a cultural dish environment, which is a plastic, and normal cells do not like to grow very well. So it's we have to understand that is only a model. Okay, what we observe and see in that model might not represent hundreds, thousands, millions of cancer patients. So this is very important. So when you are applying the running experiment, like good journals ask you to, hey, you use cell line? Okay, I don't believe if you use only one cell line. You have to use at least a couple of breast cancer cell lines to talk about what your results is about breast cancer treatment. Okay, likewise, even in brain, the immortalized cell lines are not very many in the brain neuroscience. But when you happen to use it, you have to ask yourself, is this a good model? And is this have a real implication for the ultimate goal of the cells inside the brain? Okay, so I hope you understand these questions. You always have to ask yourself the question, I hope, okay? So 3 categories of cells used in culture experiments are first, immortalized cell lines, and second, a primary cells, which is just pluck out the cells from the living body, okay? So that's why we call primary cells. And the third is about stem cells. And we will not cover stem cells here, okay? You may have to take advanced course for this. Okay, let's move on. So let me check. I know many of you have done, so raise your hands if you haven't done cell culture experiments. I just want to catch, okay, raise your hands. Who haven't done cell culture experiment? Okay, great. Yeah. Some of you have done, but many of you haven't done. So it's a good time to talk about cell culture equipment and reagents. I strongly urge you to come to my lab or anywhere the labs in third floor or Professor Oh Chang-myung's lab, because this is a basic equipment and you guys will have it's good to know what they actually mean. Okay, you can see here, typical cell culture room. This is a laminar hood, okay, one. And then this is a incubator, okay? And there are like a number of things. This is chiller, and this is a fridge, and this is a vacuum, which will be used for sucking out a medium from the cell. And 6 is people not use this more often these days, because in the past we actually reused the glass pipette by autoclaving. Now we are making it more like disposable ones, okay? So pipettes are here in this metal box, and 7 is pipette tips, okay, box. And 8 is a microscope, which is usually inverted microscope. So you have to check the condition of your cells like every day, okay? Or every other day at least, especially when you're changing your media. And 9 is it's another tool. And 10 is a centrifuge. So centrifuge, so when you have a cell suspension in in a tube, you use centrifuge because the cells are heavier than water, so that you can actually make a cell pellet to split the cell. Okay, 11 is it's a freeze. It is a cooled. You can cool and centrifuge. Okay, and 12 is a pipette made. Here you attach this pipette tips, and then so that you can have this pump to wash the cells or split the cells. And 13 is a famous 70% ethanol, which is broadly used to clean before and after. Even whatever you bring from outside to inside this laminar hood, you are supposed to spray this ethanol so to prevent any contamination. So I think I put these ones, some of the names over here. So I rarely use this, but osmometer measures the osmolarity. Okay. All right, so now let's see the real thing in picture. So here is the upper one, the CO2 incubator, and I showed you part of this as a laminar hood. So laminar hood is a place where everything is supposed to be clean. So the air is from top to outside. This is the airflow direction, and that airflow has to be filtered. So here inside, there exists a HEPA filter, which filters any kind of bacterium or like microorganisms. And then the air should come from in to out, so that, you know, everywhere there's microorganisms, even in the air too. So it doesn't suck inside, so they contaminate. You prevent the contamination. So to artificially mimic the endogenous in vivo cellular environment, I mean, even in vitro experiment, the cells are originally coming from in vivo. So we are trying to mimic and making the condition as much in vivo-like as possible. So for the cell to survive, so to supply the cell, we have to provide them oxygen, nutrients, and growth factors. Like in vivo condition, we have blood vessel to diffuse the oxygen and nutrient and even remove the waste. While we removing the waste in vitro is we just like really washing the media out and then pouring a new media. And sometimes when we have to grow the cell, we also provide growth factors, another element which is necessary to keep the cells alive. Okay, So to prevent contact from bacteria, fungi, and other microorganisms, we use this laminar hood. Jo Ji-hoon, can you guess what this is? Now you've done some in vitro experiments.
Contribution?
Oh, somehow I can't hear you, so that sounds a little strange. But anyway, I think when I see your mouth, you are saying prevent contamination, okay? So the reason how we can do it is I said laminar hood, which means the flow is laminar, which is it's making a kind of curtain with a laminar flow. Why not turbulent? Because turbulent is mixing up so that it's easier to contaminate, while laminar makes a curtain with the air flow to block, prevent the inside from this outside airs getting in through, okay? So, and then let's talk about this a little more, biosafety hood and cell culture, okay? So this is called a biosafety hood or laminar flow cabinet. And this is quite a equipment because you have to work inside. At least you put your hands, only hands over here, into here. And to prevent contamination by microorganisms, when you are not using it, we shine UV light. So turn on the UV light lamp when not used. Why? UV light has a shorter wavelength, higher photon energy. It can damage the DNA of these microorganisms. If we shine UV light onto our skin all the time, we are likely to get skin lesion, like even skin cancer, right? So that UV will destroy or not prevent these microorganisms to grow. But also very important thing is before and throughout, from time to time, and even after experiment, you are supposed to spray all surfaces with 70% ethanol. I said this many times. And while this is a lecture, but you've watched it, the TA did and Min-sung, I would strongly suggest you, if you haven't done this, talk to the people and watch. There are a number of these classmates who actually are running this cell culture experiments. You better to actually observe and watch, okay? So for example, because there are many small but important tricks which you will only learn where by doing it, okay? For example, never pass glove hands or over your bottle or cells or equipment. So what it means is, hey, let's say this is cell culture dish. I use my hand to do something. So cell culture dish, I use my hand on top. Quick, because there's some reagent I have to like pour it. So what happens? Laminar hood, the flow direction is from top to bottom. So if the dust and microorganisms and bacteria on my clothes or hands, while doing this, that can shed into your culture dish to contaminate. So you have to kind of keep thinking what can happen. And so that's why you never supposed to pass glove hand on top. So either the dish has to be covered up or you kind of carefully not to do this. So this is something like little tricks to prevent contamination. All right, so the cell culture incubator is the role, incubator is number 2 here. The role is, this is look like a refrigerator, except that this has a little different temperature and humidity and gas condition, okay, that house and store culture flasks. And this will maintain appropriate temperature and humidity. So you will find the lower bottom, there's always water. If it's drying, you are supposed to pour deionized water so that it can provide humidity. Okay, any cell, no cells can leave dry condition. which is good way to prevent coronavirus. Even bacteria with dry condition, they can't be active, right? So humidity is important, and gas concentration. Usually we use a CO2 gas to mimic endogenous condition. So what degree? Yeah, it's like our human body temperature. So the best for mammalian cells are 37 degrees. And you will watch here, like there's usually indicator about CO2 levels. That's usually 5% CO2. Okay, now I want to show you, you already probably observed some of these ones, cell culture dishes and flask and plate. So when you started, you are supposed to use the cheapest one and simple one, such as this circular plate. So this has a 60 millimeter, this has not sure. This is 100 millimeter diameter, even there are bigger one. So the good thing of this is a cheap, easy, convenient, but the downside is when you open it, the top is completely open, so it's easy to contaminate. Okay, when you move your hands around on top by mistake, it's easier to contaminate. And when you shake it, the cell media are supposed to be in the bottom, but they can go to touch here, and that's where the contamination easily occur. So that's cell culture dishes of circular. So to prevent that, we have now cell culture flasks, which looks like this, and we call it as a tea flask. I don't know where does the tea come from, but maybe this look like tea shape. But there's usually numbers out there, T25, T75, 150 to 25. This number usually means the area of the bottom where the cells are actually growing, okay? And I particularly love to use this because you see this a small narrow neck opening. Because of this, there is a much less chance of contamination when you do it. Or even when you are making it stand up, you can use it for not adherent cells, but floating or suspension cells, such as immune cells. You can actually use this one very well. But this is more like to grow the cells, expand the cell so that you can perform multiple experiments, in vitro experiments. Or you can use this one for injecting into the real body, for example, to form a tumor inside the brain. You need a lot of cells for critical mass. So you have to use a bigger T flask to make enough number of critical mass of number of cells to do something in vivo. However, for, as I said, in vitro experiments, the great advantage of in vitro experiments is actually you can have a multi-well plate, which means you can do experiments simultaneously in 6, 24, like even 96, or even 384, like simultaneously. So multi-weld plates are great ways for this in vitro experiment. But I do know sometimes people who uses a zebrafish, you can actually have put the one zebrafish in one egg, and then you can watch their development in vivo and like in vivo condition of the animal itself. Okay, so these cell culture dishes, usually the bottom of them we call substrates for cellular adherence for the cells. Like often, especially many type of cells or neurons or many cancer cells, which is origin from the epithelium, they are usually adherent cells. Adherent means attaching. They like to grow at the bottom surface, on the bottom surface. Sometimes the cells may not grow well while touching these plastics. So many transformed cell lines can grow very well, but especially when we talk about primary neurons, they don't like this. So we have to sometimes we have to try that out to coat with the surface so that the cells can attach and grow well. So we coat with some type of amino acids. Okay, so for example, lysine or ornithine. Okay, or like inside our body, we sometimes try to coat with extracellular matrix components, okay, such as, let's see, Eugene, can you guess what this is? Oh, yes, collagen. And the other, can you say, I'm watching your mouth? Okay, laminin. All right. I know, those who haven't watched a cell culture may not be familiar. Let me explain you. So lysine is an amino acid. There's a like poly-L-lysine or poly-D-lysine, like, L type and D type is coming from organic chemistry, that the lysine we are called is trying to make electrically, the cell surfaces are negatively charged, and this have a positive charge. So you coat with this surface, the cells are likely to attach better, okay? While collagen, the cells inside our body are usually touching, outside of the cell, extracellular matrix component. So collagen is sometimes you use, collagen is known for the most abundant proteins in our body. About like 30% of protein mass is collagen, okay? And laminin is a major component of cellular basement membrane, all right? So that's what, especially for primary neuronal culture. And you will find a wet lab, there are refrigerators or water bath and microscope. So refrigerator, as you all have in your home, okay, what's the temperature of refrigerator? Which is, yes, 4 degrees C Celsius incubator. And that usually, it's the same as our refrigerator. We call it short as fridge, okay? We just call fridge, okay? What does it do? We keep the cell culture media here or other reagents when not in use. It's the same as the goal of why we have a refrigerator at home, to keep our food, okay? So growth factors and antibiotics, they degrade over time, okay? But they can last for weeks if stored at 4 degrees C. For example, growth factors are, I guess this chemical proteins, and those, it's the same as your food. Your food, you put it in your like refrigerator, they can last for weeks, but you don't keep it for too long because the proteins will degrade, unless it's just pure chemical drug, which can last for years, okay? So in that aspect, you kind of have to think of this, why we are using refrigerator for culture media, because there are a lot of proteins, okay? So you want to keep it over there. And water bath. Water bath is look like this. You see 37 degrees C. So when you are pouring your cell culture media onto the cell to quickly warm the cell culture media using this water bath before adding to cell culture flask and plates, because that's what the cells are originally happy, okay? Because even the cell incubator was 37 degrees. So it's like, you wash with the culture media with the same temperature. However, I can say sometimes when you use a cancer cell lines, they are so strong that you don't even warm the cell culture media. I've done many, many times for this. But, primary cells and more like subtle cells, it's better to use this warm bath to warm the cell culture media, because the fragile cells may die or change its phenotype because of the cold temperature. Usually we keep the cell culture media at 4 degrees. You don't pour this one onto very important or primary cells. Okay. And microscope is you have to watch with your eye. So routine observation of cell culture flasks and plates to inspect the health and the amount of cells like growing on the cell culture flask. Okay, that's called, let me see. Okay. 한진욱, yeah,
Confluency.
can you guess?
Uh,
I can see your
confluency.
mouth.
Confluency.
Okay, yeah, confluency of cells. Okay, this is a terminology you have to know when you are performing cell culture. The reason is, hey, this is an example. So let's say when you have plated your cells initially, let's say 10%, the cells, if you pour or growth factor together, they will grow in number, okay? And of course, so a couple of days later, you see, you define, hey, in terms of area, how much the cells are occupying, like 30%, 50%, 90%. When you see this 90%, this is not a good time because you are supposed to already split the cells, not reaching 90 or even 100%. Okay, so here you can see the definition of confluency is very easy. The percentage of the surface of the plate covered by adherent cells. You know, we are talking about adherent cells, cells that grow touching on top of this flat cell culture plate. So cells has to be kept in healthy and growing state. That has to be subcultured. What I mean is when you grow the cells in this flask, the goal is to make enough number of cells so that you can run experiments, okay? Multiple experiments. So typically what happened is you grow the cells before reaching to 90%, what you do is you have to take out the cells because there are too many cells in one dish. And you actually have to suspend the cell. And let's say there's like 3 million cells inside, then typically you divide 2 or 3 times. So 3 million, you take out 1/3, 1/3, 1/3, and then you pour that cells into another new 3 culture dishes. So that we call subculture. Then you start with 30% or 10% again, and you grow them. So it's like exponential usually growth. But when I say growing state, so this process we call subculture, or we call passages. So this is passage 1, and you subculture the next generation, that's a passage number 2. So over like years, you know, it's a very good idea for you when you first obtain the cells, let's say from the company called the ATCC, or you got it from your collaborator, or you make it from your own animal primary, write that down the number of passage, because this can be very important later on. Why? Especially primary cells, when you do this subculture over and over, the cell phenotype may change. And you have to write that down and then report it in your paper or thesis, because otherwise, you know, later people try to reproduce your results and they got different results. And they can accuse you, hey, you guys published this. I waste my 5 years of my PhD just to reproduce what you have published. There's nothing like that. You don't want to be in that condition, okay? So that's very important to understand primary cells can change its phenotype over the number of passes. That's why at least, internal consistency and information, you better to keep track of the passage number. And in growing state is, what does that mean is, when the cells reach this 90 or even higher, these cells who are contact cells usually have a tendency not to grow faster at over this condition, because you see there's no room. So when they touch each other, there's internal mechanism to inhibit the growth. We call it contact inhibition. Then these cells may not grow very well. So the point is, before reaching this 90% or higher, you are better to subculture, okay? 70 to 90, even between 50 to 90%, when you find that, oh, you better to subculture, okay? It's time to passage cells. So why we don't want, the most effective way is we wait until confluent. Yes, the number is high because cells tend to inhibit each other's growth. I said contact inhibition. And what happens? Because there are a lot of number of cells, they quickly drain nutrients. That means you have to change the culture media supposedly even faster, okay? And even because of that, the pH of the media will change, and that turns the color of your media from pink to yellowish, because the cell culture media usually have pH indicator, which shows you, hey, something's changing. So it's time to change your media. Okay, so I'm going to talk about culture media. What's the role of growth media? Like inside our body, our cells are always showered by nutrients and oxygen, and even the vessels will collect the CO2 and waste, okay? So culture media is to supply nutrients such as amino acids and vitamins, and source of energy, glucose, for the cells. So what are the requirements of culture media? You have to think it like inside the body, the fluid inside the body, such as a blood plasma or interstitial fluid, which is showering together with all the cells all the time. So you have to have same concentration of solute molecules as inside the cells. So that we call as isotonic condition. Otherwise, the cell will either, like osmolarity, osmosis. The cells either like burst or shrink. You don't want that condition. And pH, you have to meet the pH, okay? We have buffer to maintain a compatible pH. Okay, bicarbonate buffer is being used to maintain physiological pH and provide nutritional benefits. So pH is usually this is a good number to remember, about 7.4, which is our physiological pH. And sometimes you provide the survival signal or growth. So promote survival through undefined growth factor, many growth factors, and hormones and proteins such as PDGF, insulin, transferrin, many kind of proteins. Sometimes you add this called the serum, okay? The definition of serum is actually pretty easy. What it means is, you have a blood, you draw blood, it's red, but if you centrifuge red blood cells down and you have a yellowish liquid that we call blood plasma, okay? But it contains a clotting factor. So by removing clotting factor in the plasma, we call it serum. This contains a lot of growth factors and good things. However, you have to remember it's not undefined. It's undefined growth factors. So you don't exactly know. And each serum, such as bovine, like BSA, bovine Okay, calf or bovine serum, you have to understand that's undefined. So sometimes for careful in vitro experiments, you have to consider the use of this serum carefully. So that's why serum-free chemically defined supplements are sometimes used, okay? Because they have a known formulation of survival factors. Example is N2 or B27 called neurobasal media. So neurobasal media is providing optimum amino acids and nutrients to culture neurons, okay? So that is sometimes important compared to this serum. All right, immortalized cell lines, which is a, sorry, this is a table, like this is more general than neuroscience only. So I'm gonna go just give you a point because you will hear this while in this field a lot of time. 3T3 cells are fibroblasts, which is a fiber making cells. You know, when you have a cut or injury, there's scar formation made by fibroblast because it generates collagen. Okay, it has contact inhibition. HeLa cells, a famous cancer cell line from a cervical cancer. You probably hear COS cell line and HEC cell, 293 cell, and CHO cell. The original name is good to hear once, but the point is of COS cell and HEC cell is it has efficiently transfected, which means I want to make a cell a genetic modification, overexpressing some kind of genes or suppressing a specific genes, people use this one, okay? CHO cell is useful for stable gene expression and high protein yields for biochemical assays. All right, so there are some like neuron-derived like cells, mouse neuroblastoma, and these cells. Okay, so what do I mean by immortalized cell lines? I said cell lines. So some cells, the previous cells, PC12 cells, we add nerve growth factor, then the cell looks like a neuron, and it's immortal. So that's easy to work with in neuroscience, right? So immortal cell lines can be cultured over generation, and immortal already means it's like tumor-like cells because the cell keep growing, it forms a tumor mass that do not stop dividing. And this is special cell that's artificially manipulated to proliferate indefinitely. You see here the term of artificially manipulated, which means you gotta be very careful to draw conclusion from this cell line experiment. It can give some insight, but do not trust the conclusion will be applicable for any situation, okay? And for these cells, we transfer a fraction of multiplying cells into new dishes to provide space for continuing proliferation. I call it passaging, or another one, because we split the cell, the number of cells we call splitting, okay? Okay, so then why using immortalized cell lines? We still use it, provide standard cell types that are well characterized for reproducible results. Yeah, one way of think of a cell line is, hey, I've done this research results and we can compare with others, because primary cells could be much more diverse because it depends on the skill very well, and there are mixture of multiple cell types in vivo condition. So it's more easier to reproduce so that scientific community can share the results and reproduce each other. Homogeneous and genetically identical population for consistent results. I put in theory, the reason is it's a cell line, so it's supposed to be the same. The reality is, if you watch it carefully, it's not perfectly homogeneous. That you have to understand, okay? All right, easier to culture than primary culture. Of course, primary culture is more difficult. So cell lines are growing more robustly and doesn't require extraction of cells from a living animal. And it's immortal, so it grows quick and continuously. What about primary cell? They don't continuously grow, okay? So when some type of study, you have to extract large amount of proteins for biochemical assay, such as finding out the amount of protein Western blot, or for with the antibody assay, you do need a lot of protein, which this can provide a lot because it keeps growing. While primary cells, you can collect only tiny amounts. So sometimes you have to use many animals to have enough number of cells for your study. We can create cell line to continue to express a gene of interest. Yeah, sometimes you want to manipulate the gene, then you need cell line because they keep growing, while primary cells are usually one-time use, okay? So that you can add fluorescent tagging for specific proteins of interest or mutant, generate a mutant protein. All right, so then what are the limitations? I already alluded. They are not normal, okay? Dividing indefinitely means it's already abnormal. It's like cancer-like, okay? Not usually found in vivo cell type, of course. So it's not normal, so you have to suspect the results, okay? You have to limit your conclusion. And as I said, cell characteristics, even cell line can change over passage. So be careful and always compare with the published results, baseline first before you draw or further moving, doing more experiments, because you have to suspect your cell line first, okay? Because otherwise, if the phenotype has already changed, you may not be able to publish. So that's something you have to remember. Immortal cell lines of neuronal origin, not a lot, to study properties unique to neurons. For example, to investigate process that occur during differentiation in neurons, such as axon selection, axonal guidance, and growth. So there are only a few available. PC12 neuro2A, so from rat, from adrenal gland tumor. You see it's from tumor. And neuro2A is a mouse neuroblastoma cell line. Hey, you see, it's a blastoma. Oma means tumor. So it has to be from tumor to become cell line, okay, to become immortalized. Primary cell and tissue culture is what you've done in the demo. So extracting brain, you take out the brain, and then you sometimes section of brain of interest or dissect the brain region. And or sometimes you have a hamburger steak of brain. It's like steak. So slice culture. Sometimes we culture this way, or we take out only of our interest as keeping that part called explant culture. Or we just chop that out and enzymatically dissociate. So dissociated culture. So these are three types of primary cell and tissue culture. So primary is derived from tissue removed directly from a living animal. You kill the animal, you take out the cell, okay? Why we do this job? To directly investigate cells of interest in a carefully controlled. Because in vivo study is very complicated. So you sometimes, before in vivo, you do this primary, because you can do controlled in vitro environment. I'm talking about primary cells, but we are running in vitro experiment. So what are the limitations we have to keep in mind? Yeah, primary cells are not cell line. They don't live forever, okay? They have limited lifetime. And even the age of the animal matters, okay? So animal source influence the health and robustness of cell culture. You see why primary cells are more complicated? You have to even match the age of the animal source, okay? And when you take out the cells of your interest, they will never be alone, okay? The populations of primary cells compared to immortal cell line, they are supposed to be heterogeneous. For example, you are interested in this primary neuron. What you've done is trigeminal ganglion neuron, but you actually have together with a glial cells together. It's heterogeneous. So it's a little bit more difficult to do clean experiments compared to neuronal origin cell lines. So I gave you some comparison chart of Immortal and primary culture. Now I think you understand clearly Immortal, easy to use, homogeneous, well characterized to compare between groups over the world, between scientists, and you can create stable cell lines expressing genes of interest. You can do this experiment. While the results may not have the same of the neuron of interest. While primary is more relevant, cell physiology and brain neuronal circuitry, while the result is more hard to interpret because it's heterogeneous population with high variability. So slice culture and explant culture and dissociate cultures, I think I'm going to go over quickly for all of these. Brain slice is keeping structure and organization of brain in a relatively thick, about up to 400 micron. Why 400 micron? Now you take out the brain as a slice. Brain has a blood vessel, but you take out no vessel. So you have to provide oxygen nutrient from the water bath. Then there's solid diffusion in mechanical point. Diffusion can only reach up to several hundred micron. If it's thicker, you will find the inside the cells will die out, okay? And how to cut it? We use vibratome. Tome means cut. So vibratome device, we are using it. And why are we doing this? Because I'm interested in, let's say, hippocampus, a specific area related to learning and memory. You can access it from the outside because it's at least 1 millimeter down from the surface of brain. So access, and we have to see deep subcortical structures, such as hippocampus, thalamus, that can be very difficult to access, intact in vivo. Okay, so types of slice, 2 ways. You cut it in the morning and then you keep it to run your experiment for several hours and throw it out. That's most of the time. Case it's called acute culture. Use it immediately for short-term electrophysiology experiment. The quality of slice matters over 90%, some of my colleagues said. So that requires a good skill to keep the cell slice alive. Another way is even you can culture that slice of steak or brain. Okay, that's called organotypic, which is brain as an organ. We use that organotypic slice with the culture. So we can maintain over multiple days. Then it can be useful for structural morphological changes. For example, neuronal migration. Some cells migrate over hours and days. It's great to use this, or axon outgrowth or synapse formation. Okay. The importance of this slice requiring air fluid interface to regulate gas exchange throughout the slide. So this is when you run the experiment. I'm saying this, this is not easy actually. So to get familiarized, you need the best ways to go to the lab who do this over years or decades and learn the tricks. This is better to actually see it. This is brain harvested. You cut this chop. So the way you cut with the vibratome, now you have this slice of cut. And you have to keep this media with oxygen, okay, and nutrient. And you have to keep it in ice, all right? So this is called a vibratome. It's an instrument to cut thin slice, similar to microtome, but vibrating blade. So it works like this, like this. It proceeds, but it vibrates, okay? And the quality is very important. There are parameters you have to actually choose. Okay, explant culture is not slice, but part of the brain regions you are interested. So intact fragments with the same mixture of cell types. So it's not pure cell, single cells, it's a tissue fragment brain. not necessarily preserve precise orientation of the nervous system, but you can submerge in the media and hey, there's a control and there's a chemical to see if the role of this chemical for this, you see more of this growth in this direction. So this is a great way of a in vitro primary cell culture study. Okay, so co-culture assays studying neurite outgrowth. You see neurite outgrowth and neuronal migration. Explant culture is a good way. So for example, axonal growth of inorganic organotypic neural culture using explant. For example, spinal cord DRG stands for dorsal root ganglion. You don't have to remember for now, but you can cut this and then you can culture, and you saw that this neurite growth, you can do, take this image, and using image J, you can process and quantitative analysis so that neuronal branches, you can quantify them. And then you can study between control and experimental group. Okay, dissociative cell culture. Okay, brain tumor, for example, there are cells you dissociate and you proliferate and dissociate and you proliferate and establishing cell lines, and you can differentiate into neurons. All right, so neural tissue can be separated into individuals. So in the end, you make individual cells growing 2D or 3D substrate. Then you choose a specific region of brain. For example, what you did last time is a trigeminal ganglion, okay? Can be micro dissect and to isolate. Then you have a specific neuronal subtypes. In your case, trigeminal neurons. Then you can use mechanically chopping or enzyme to use to digest within a suspension to separate individual cells, okay? And with the proper growth factors and care, you can maintain those cells for weeks so that you can study them, how they grow. So for example, dissociative cell culture, actually this study has been done in our lab, and what you've done is related to this. Study neurite outgrowth, how well the neurites are growing, or synapse formation, or even electrophysiological studies. What are the limitations? You dissociate, you lose the organization and connectivity that are critical to in vivo functions. So because you are doing in vitro study, and small quantities of cells can be difficult to perform. They are too tiny for biochemical experiments. And what are biochemical experiments? You have a tissue chunk, you grind them, you run the study to find out the biochemical aspects. It's difficult for that. And heterogeneous, of course, often mixtures of both neurons and glia, okay, which is non-neurons in the brain that responds to different neurotransmitters. So identifying individual population of cells can be difficult. You see the limitation here, but the advantages are, you can reach these cells in vivo very easily. So let me conclude. Growing cells and tissues outside of a living organism for in vitro. So greater access and control over the cellular environment compared to in vivo condition. And in vitro study, you can investigate cellular molecular mechanisms of nervous system function in more detail because you have a greater control. And this provides many informative results, while you have to be careful interpretation, or you have to confirm similar things happening in vivo when you are drawing a more bigger conclusion, okay? So ultimately, this knowledge must be tested in intact nervous system. And you did the demo last week. And so I know those who have done many of these could be easy, but there are new people. So culture equipment and reagents, we need a special equipment and reagents and culture media. And sometimes we use immortalized cell lines by growing them, splitting, passaging, but we have to be understanding the pros and cons of the cell lines. while in neuroscience, we sometimes use a primary cell and tissue culture with a slice culture to preserve the connections and orientation, and sometimes explant culture to take out only regions of interest in the brain and see the behavior, or sometimes completely take out that part and single cells, and we study those single neurons, dissociate cell culture. Okay, this is the end, and next time visualizing neural structures.